The iodine clock reaction is a classical chemical clock demonstration experiment to display chemical kinetics in action. It was discovered by Hans Heinrich Landolt in 1886. Two colorless solutions are mixed and at first there is no visible reaction. After a short time delay, the liquid suddenly turns to a shade of dark blue. The iodine clock reaction exists in several variations. In some variations, the solution will repeatedly cycle from colorless to blue and back to colorless, until the reagents are depleted. Hydrogen peroxide variation. This reaction starts from a solution of hydrogen peroxide with sulfuric acid. To this is added a solution containing potassium iodide, sodium thiosulfate, and starch. There are two reactions occurring in the solution. In the first, slow reaction, iodine is produced, H2O2 plus 2E plus 2 hours plus I2 plus 2 hours 2O. In the second, fast reaction, iodine is reconverted to two iodide ions by the thiosulfate, 2S2O32A plus I2OS4062A plus 2E. After some time the solution always changes color to a very dark blue, almost black. When the solutions are mixed, the second reaction causes the terdide ion to be consumed much faster than it is generated, and only a small amount of terdide is present in the dynamic equilibrium. Once the thiosulfate ion has been exhausted, this reaction stops and the blue color caused by the terdide starch complex appears. Anything that accelerates the first reaction will shorten the time until the solution changes color. Decreasing the pH, or increasing the concentration of iodide or hydrogen peroxide will shorten the time. Adding more thiosulfate will have the opposite effect. It will take longer for the blue color to appear. Iodate variation, an alternative protocol uses a solution of iodate ion to which an acidified solution of sodium bisulfite is added. In this protocol, iodide ion is generated by the following slow reaction between the iodate and bisulfite, IO3A plus 3HSO3A E plus 3HSO4A. This is the rate-determining step. The iodate in excess will oxidize the iodide generated above to form iodine, IO3A plus 5E plus 6 hours plus a 3I2 plus 3 hours 2O. However, the iodine is reduced immediately back to iodide by the bisulfite, I2 plus HSO3A plus H2OA2E plus HSO4A plus 2 hours plus. When the bisulfite is fully consumed, the iodine will survive to form the dark blue complex with starch. Persulfate variation. This clock reaction uses sodium, potassium or ammonium persulfate to oxidize iodide ions to iodine. Sodium thiosulfate is used to reduce iodine back to iodide before the iodine can complex with the starch to form the characteristic blue-black color. Iodine is generated, 2E plus S2O82A I2 plus 2SO42A, and is then removed, I2 plus 2S2O32A A2E plus S4062A. Once all the thiosulfate is consumed, the iodine may form a complex with the starch. Potassium persulfate is less soluble while ammonium persulfate has a higher solubility and is used instead in the reaction described in examples from Oxford University. Chlorate variation. An experimental iodine clock sequence has also been established for a system consisting of iodine potassium iodide, sodium chlorate and perchloric acid that takes place through the following reactions. Terdide is present in equilibrium with iodide anion in molecular iodine, I3A plus I2, chlorate ion oxidizes iodide ion to hypoiodous acid and chlorous acid in slow and rate-determining step, ClO3A plus E plus 2 hours plus Ahoy plus HClO2. Chlorate consumption is accelerated by reaction of hypoiodous acid to iodous acid and more chlorous acid, ClO3A plus Hoy plus H plus HiO2 plus HClO2. More autocatalysis when newly generated iodous acid also converts chlorate in the fastest reaction step, ClO3A plus HiO2 and IO3A plus HClO2. In this clock the induction period is the time it takes for autocatalytic process to start, after which the concentration of free iodine falls rapidly as observed by UV vis spectroscopy. See also, clock reaction, Old Nassau reaction. References. External links: hydrogen peroxide variation, potassium iodate variation, potassium iodate variation 2, sodium bisulfite variation with a high-speed camera.